Well, thank you very much. It's a pleasure to be here and see people who are becoming old friends. There are quite a number of people here who I've seen at meetings recently. So I'll be talking about electromagnetic fields and how they impact the cells of our bodies. And what I'll be arguing is that they work via the activation of what are known as voltage-gated calcium channels, and that the effects of that activation then lead to many, many different impacts on health. And so, so what I think you're all familiar with is that there's been a great puzzle about these EMFs, and that is these EMFs are composed of low energy photons, with the energy per photon too low to influence the chemistry of our bodies. And so the question is, how can they influence our biology by anything other than heating, by thermal effects? And as you probably know, the safety standards assume that they cannot, that the only thermal effects need be considered. There cannot be biological effects, they claim, other than by heating. But we know that there are thousands of studies in the scientific literature that say that cannot be true, that there are many different biological effects that occur at levels well within safety standards, and which should not be there then if the safety standards have any scientific merit whatsoever. So, but that, of course, leaves open the question of how this thing works. And what's true is that there are 26 different studies on EMF effects. And these are of a variety of different EMFs, the microwave and radio frequency EMFs, but also extremely low frequency EMFs and even static electrical fields and static magnetic fields. All of these things can act by activating voltage-gated calcium channels. And the key observation is that these effects can be blocked by calcium channel blockers, which are drugs which are specific for blocking these voltage-gated calcium channels. And when these voltage-gated calcium channels, and I abbreviate these VGCCs, are activated, what happens is that they open up a channel in the plasma membrane of the cells, and you get calcium flowing into the cell. It's the excess calcium in the cell that's responsible for all or almost all of the biological effects. And so basically, that's sort of a summary of a lot of the things that I'm going to say. This observation was initially made in this paper that I published in 2013 in Journal of Cellular and Molecular Medicine, and we'll talk about that a bit later. Now there's another issue that I wanted to introduce here, which is another part of this puzzle, and that is it's been known, actually I say for over 30 years, actually it's been known for over 40 years, that pulsed electromagnetic fields are often much more biologically active than non-pulsed fields, even when they're producing the same heating or even less heating. So this is obviously inconsistent with any notion that heating is what's going on here, and it leaves another issue, and that is that essentially all of the wireless communication devices communicate via pulsations. They use different pulsation patterns, and so we don't know enough about this to really predict how one pulsation pattern is going to compare with another in terms of biological effects. And so this is another difficult area which impinges on your interests. How do you determine how biologically active particular fields are? You cannot simply look at the intensity of the field as a measure of the effectiveness in biology. Okay, so how do we make sense of this? Well, as I said before, these fields act by activating these voltage-gated calcium channels, and the central evidence that shows this is the fact that you can block these effects with different calcium channel blockers. And there are a grand total of 5 different calcium channel blockers that have been used in these studies, each of which is thought to be highly specific for blocking the calcium channel, the VGCCs. And so, you know, it's not just one channel blocker has been studied, but 5 different ones. They all work on different sites. They all have different structures, but they all have this common effect. They all block the VGCCs. Okay, so, and so I've got a table here, and this actually only includes 23 of the 26 studies. But what you'll see here is there are a number of different types of fields. Most of them work via what are called L-type VGCCs. And I don't really have time to talk about what the difference is between these different types. But you'll see here that you have a variety of different fields, including things like the NMR thing on the bottom and the pulsed magnetic fields on the top. Now, those are both microwave frequency effects, but then there are also extremely low frequency effects, and including the 50 hertz ones that are listed here. And even static magnetic fields work via this mechanism. And so, oops, let's see, am I going? Okay. And so, again, you also get nanosecond pulses, so very short pulses of microwave frequency fields can activate these channels. And so you have a couple of there on the nanosecond pulses, more 50 hertz and 60 hertz. And then down at the bottom, another one that's working on a different type of channel, the N-type channel, and working at, you know, in the about 1 gigahertz range, so microwave frequency, and so forth. So, oops, and there's still others. And so so this is a general pattern, and of course, the main concern we've had with health effects have been due to the microwave frequency fields because our exposures have been going up at extraordinary pace of those. And so that's the main concern. But there are also concerns from the extremely low frequency fields from the wiring and from in our house and from you know, high voltage power lines and so forth. And so those are all things that I think you are interested in. So I won't say much about extremely low frequency fields, but I think they work very much the same way. Okay, so now when you look at this, this is a slide that was taken from one of Professor Annette Dolphin's papers. And on the top of it, you can see a structure which is a model of the structure for the VGCCs. And in fact, for all the different VGCCs, there are 10 major types that are listed here. And what you'll see here, those cylinders are going across the plasma membrane. Okay, so that structure there that they're in is the plasma membrane. And let me just say, we actually know a lot more about how this structure actually is, and it's not really quite this way, but this is a way of thinking about it. And if you look at this, there are 4 different domains there that you can see that look very, very similar to each other, right? There's, you know, Roman numeral 1, Roman numeral 2, 3, and 4. And each of them has 6 different alpha helices. I've got one.
This is better.
Okay. What did I do? Oh. Okay. So each of them has 6 different alpha helices. Okay, so we have these 6, and what you see is the 4th helix, which is in a different color here, has 5 pluses on it. Okay, those are positive charges. Okay. So there are 5 positive charges here and there and there and there. Those make up what's called a voltage sensor. This is the structure which detects electrical effects. Okay? These these four alpha helices that are in orange here are represent the voltage sensor. Okay, so the voltage sensor in the normal physiology detects electrical changes across the plasma membrane. And what I'm going to argue is that these are the same structures that are detecting the electrical effects of these fields. Okay? So the fields can produce electrical forces on these positive charges, and that's the central mechanism by which these fields activate these channels and produce biological effects. Okay? So, so the conclusion from this is that these various kinds of EMFs act biologically by activating the VGCCs, and that there are, and this is further supported by literally hundreds of studies showing that, and this is specifically low-intensity microwave EMF exposures, are followed by changes in calcium fluxes and also changes in calcium signaling. And these can each be explained as being a consequence of the VGCC activation. Okay? So there's a much larger literature then that argues for VGCC activation as a response. And so we know that there are changes in calcium fluxes and calcium signaling. We also know that there are increases in oxidative stress in response, and I'll talk to you about how that's produced. And interestingly, Dimitris Panagopoulos has proposed from biophysical modeling that EMFs can act by activating these types of voltage-gated channels. So, so there are a number of things that point in this direction. And in addition to that, there was a very important study that was published by Arthur Pilla. And what he showed was you have cells in culture, and you expose those cells to a pulsed microwave field. And when you do that, you get a calcium calmodulin dependent increase in nitric oxide, all happening in less than 5 seconds. Okay? So this is almost instantaneous. This argues strongly that the effect on the VGCCs is a direct effect because it occurs incredibly rapidly. When you get an increase in intracellular calcium, that in turn activates two enzymes that make nitric oxide, which are calcium-dependent enzymes, and they then produce this nitric oxide, and all this stuff occurs in less than 5 seconds. So you can measure it almost instantaneously. I'm going to come back to this a little bit right at the end because I think this kind of methodology may be very important in terms of looking at real biological effects of these fields at the level of cells in culture. Okay, and so now there are other things that point to the VGCCs as key targets. And it's interesting that, and this came out of 2 reviews on the VGCCs, one of them by William Catterall, who's an expert at the University of Washington on this, and another one from Dr. Chen, who's another world-class expert on the VGCCs. And both of them say that when you have electrical effects in the cell, they work to produce chemical changes, as far as we can tell, exclusively via activation of these VGCCs. Okay? This is the mechanism for converting electrical effects into chemical changes in the body. Okay? And so it shouldn't be surprising, really, that these things are that the EMFs are working through this mechanism as well. I mean, so what we're looking at here is a whole lot of different things that are all coming in and pointing in the same direction. Okay, so recently it's become apparent that that there are also some channels in plants which appear to be the main target of the EMFs in plants. And I call them VGCC-like channels, although they actually have significant differences from the structure of the VGCCs. But what is true is there are also calcium channels in the plasma membrane, and what's also true is they have a voltage sensor, which is very similar to the voltage sensor in the animal channels. So again, I think that the voltage sensor is the target. That seems to be the universal pattern here. And and so, and I really don't have time to talk about all of these things, but basically there are a number of types of things that argue that this is a common thing in plants, including the fact that many of the effects that occur in plants, many of the physiological changes that occur in plants following EMF exposures, and these are microwave exposures that have been studied, produce changes in calcium signaling. And calcium signaling is very important in plants. It's also very important in us, in animals. And so many of the physiological changes that you see in plants are produced by this excessive calcium in the cell. So it's kind of interesting that and perhaps a bit surprising that this seems to be the same mechanism, or at least very similar mechanism. Now, there's one difference in the plants that I just want to point out, and that is the channels. Instead of having the peptides that form those channels, instead of having 4 domains, as you saw before, have 2, okay? So there's this domain and there's that domain. It's the 4th helix in each of them, again, that has the positive charges in it that make up the voltage sensor. But in plants, these are thought to act as dimers. That is, you have one peptide like this, and then you have another identical peptide forming the channel. And so you still have 4 domains in the actual channel, but they're on 2 different polypeptides rather than on 1. And so there are differences, but there are also many important similarities. So, okay, so this is another figure that I stole from somewhere. I can't remember where. I'm sorry. I should give credit for this one. This was when they thought there were only 4 positive charges on the voltage on each voltage sensor helix, but now we know there are 5. And but it's a nicer figure in other ways. And here we're back to the Annette Dolphin one, which I showed you before. And this is blown up. Unfortunately, it blows up the blurriness as well. Okay, so that's basically where we are, and I'm going to come back to that in that concept, that structure concept, in this discussion. So now, as you probably know, advocates of current safety standards claim to this day that there's no biophysically viable mechanism for these weak field EMFs to produce non-thermal effects in the bodies. And one of the papers that's often cited is this Shepard et al. Paper, which was, for instance, cited by the WHO. Okay, claiming that there cannot be a mechanism. And the basic argument that they use, and it's a substantive argument that has to be dealt with, is that while they acknowledge that the EMFs can exert forces on charge groups, and therefore can, at least in principle, produce biological effects, they argue that these weak fields are too weak really to do very much. They just don't have the energy, okay? And so that's important. They also argue that at body temperature, there are thermal effects. Things are moving around, right? And randomly, there's thermal motion of groups. And so they also argue that the thermal motion would swamp out any effect of these fields, okay? And so And so basically they have two arguments here that argue that, oh, that can't be an effect. Let me just say, we know that can't possibly be right because of all these studies, okay? But then the question is, why is it not right? And why, how can we understand this? Okay, so let's see. Okay, so the 4 alpha helices, each containing 5 positively charged groups that we just talked about, they act collectively as the voltage sensor, and they open the channel when they're all pushed or pulled in the same direction at the same time. Okay, so basically, and the fields can do that, and the changes in the charge across the plasma membrane can do that. But it has to be directional, and it has to be at the same time in order for the channel to open and allow calcium to flow into the cell. Now, thermal motions are random in 3 dimensions. So they're essentially never going to be all pushing these things in the same direction at the same time, because they're all going to be going, in 3 dimensions, pushing and pulling in various directions. So thermal motion clearly is very different from the coordinated forces of these fields. Okay, so that's one thing that's important that argues against the Shepard et al. Argument. And let me say other people have made a similar argument to that as well. And but there, in addition, 2 other things which are very important. And let me say I published on all of this in a paper that just came out a few weeks ago. So one effect is that the plasma membrane is electrically non-conductive, whereas the biological fluids, both within the cell and outside the cell, are highly conductive because, of course, water is polar, and also because there are lots of dissolved salts that give charged groups that can move around and conduct very well. Okay, so you have the plasma membrane, which is electrically non-conductive. Now, as a consequence of that, the voltage gradient across the plasma membrane at each point in time, and let me say this is going to vary from 1 nanosecond to another because the fields are oscillating all the time, of course. And the voltage gradient is going to be highly concentrated across the plasma membrane. And in fact, in the Shepard et al. Article, they estimate that there's about a 3,000 times concentration of the charge across the plasma membrane. What does that tell you? It tells you, for that reason alone, the forces on those charged groups is multiplied by a factor of 3,000 over what it would be if those charged groups are sitting out in the aqueous phase of the cell, where most charged groups occur in the cell. Okay, so that's very important. We've got a factor of 3,000, and there's another factor that comes in here. Let me just say, all of this comes out of the physics, okay? It all comes out of physics. It's not even biophysics. Okay, so there's something called Coulomb's law, which predicts how forces are going to, how electrical fields are going to influence the forces on charged groups. And one of the parts of Coulomb's law says that the forces on charged groups is inversely proportional to what's called the dielectric constant of the medium that they're in, okay? And so the dielectric constant of the lipid in the membrane is about 1/120th of the dielectric constant of these biological fluids. So what that tells you is that you're going to have another factor of 120 based on the dielectric constant that's going to increase the force of these fields on those charged groups that make up the voltage sensor. Okay. Now, so we've got. So what I did was I did a calculation then, and this is a rough calculation. Let me say this is not to be taken to be very precise, but that the force on the voltage sensor is about 7.2 million times higher than the force on single charge groups that are elsewhere in the cell. Okay? So you have a factor of 3,000, that's the concentration of the field across the plasma membrane. You have a factor of 120, that's the effect of the dielectric constant. And then you have the fact that you've got 20 different charge groups that make up the voltage sensor, okay? So you multiply those together and you get 7.2 million, okay? Now, these things are going to you know, this is not sort of a figure that I argue is fixed in concrete. There are a lot of different factors that are going to affect what the actual number is for a particular VGCC in a particular situation. So, you know, but what this clearly shows is that, that the levels of exposure that can activate these VGCCs is vastly lower than those needed to produce substantial heating. Now, why do I say that? Because heating is produced by the movement of charge groups, okay? That's what you do when if you heat food in a microwave oven. Charge groups joggle around, okay? And so here you've got forces that are like something like 7.2 million times higher. You know, so the notion that you look at heating is totally ridiculous, completely without scientific merit. And we now know why, okay? Now, you know, now there are a lot of things that are going to affect that figure, okay? And so I just want to mention a few of them. One is you have different subunits. You know, I talked about the fact there are 10 different main forms of these VGCCs. They have different alpha-1 subunits. The alpha-1 subunit is the one that encodes that peptide that we just talked about, the one with the 4 domains in it, and that also has the actual channel in it. And so, you know, so there will be variation. They're not all going to be the same. There are other subunits that also vary in their structure, that also influence how easily activated these things can be. There are various regulatory factors which occur. There's protein phosphorylation, and there's also G-protein interactions. And I think that protein phosphorylation probably has a substantial role in EHS, which has been talked about at this meeting. And there are also specific field variations that we talked about, you know, pulsation patterns. They're what are called window effects. We know that in quite a number of situations, there are specific intensities of exposure that produce maximum biological effects. And when you go either lower or higher in intensity, the biology drops off way off, okay? So this notion that, you know, the higher the intensity, that you always get a higher effect, we know that there are many exceptions to that, okay? So there are lots of things, and of course this makes your jobs much more difficult, right? Because you can't make simple predictions based on intensities of what's going to happen in terms of the biology. So anyway, so these various factors in conjunction with each other make the effects reported from even the most extremely low field levels biophysically plausible. You know, and of course this needs to be tested empirically, and it can be. But, you know, we're still in the early stages. Let me just say I'm quote-unquote retired, so I'm not going to be doing the testing. But I would like to, I'll be happy to work collaboratively with people who can do that. And anyway, so, okay. Okay, so in summary, a central role of VGCC activation in response to low-level EMFs is shown by, first of all, 26 different studies showing you can block or greatly lower effects with the calcium channel blockers. In each of these studies, and this is something I haven't talked to you about, where you block or greatly lower one effect, when people looked at more than one effect, they were also blocked or greatly lowered, okay? So that argues that there are lots of effects that go through this mechanism, not just a few, okay? And And as I mentioned before, there are hundreds of studies showing changes in calcium fluxes and calcium signaling that are all consistent with VGCC activation. Arthur Pilla did a very important experiment showing that you get essentially instantaneous increase in both intracellular calcium and following that, presumably nitric oxide, which all occurs in less than 5 seconds. VGC activation has a universal or near universal role in converting electrical signals to chemical changes in the body. The low-level EMFs activate VGCC light channels in plants containing a very similar voltage sensor and allowing calcium then to flow into the plant cell. And the properties of the voltage sensor of the VGCCs predicts that voltage sensor is exquisitely sensitive to these low-intensity fields. And therefore, the argument that there cannot be a biophysical mechanism simply does not apply to the voltage sensor. Okay? So there can be no question that VGCCs are the major, perhaps the only targets of low-intensity EMFs in the body. And then the question is, well, how do they produce biological effects? Okay, so when you activate these, what happens in the biology? Okay, so a lot of this is shown now on this slide, and what you can see here is, first of all, as we've already said, microwave and lower frequency EMFs act to activate these VGCCs, and then they produce increases in intracellular calcium. Intracellular calcium is designated Ca2+I. Okay, so that's a designation for calcium in the cytoplasm of the cell. And that in turn can produce an increase in nitric oxide, as we've already said. Now, it turns out that therapeutic effects, and there are real therapeutic effects of these fields, are produced through the nitric oxide signaling pathway. So nitric oxide stimulates the production of cyclic GMP, cGMP, which then stimulates a protein kinase, sometimes called the G kinase. And that then produces therapy. There are real therapeutic effects that occur across the top of this figure. Okay, and let me say, I argued for that mechanism in my 2013 paper that I told you about before. And interestingly, Arthur Pilla, who's a real expert on this, he's been working on this for over 40 years, had argued for a very similar mechanism at almost identical time in a paper that he published in 2013. So there's very strong evidence for this pathway being the mechanism of therapy. Okay. Now, obviously, what we're most interested in, most concerned about, is not therapy, but the pathophysiological effects. And so, I'll give you some evidence for this. But basically, we think that the pathophysiological effects are produced through this peroxynitrite pathway. So one thing, and also through excessive calcium signaling of other types. Okay. So, an increase in intracellular calcium not only increases nitric oxide over here, NO, but it also increases superoxide over here, both in the mitochondrion and outside the mitochondrion. And these two react with each other. NO reacts with superoxide to form peroxynitrite, which is ONOO-. That's the chemical structure of it. And peroxynitrite can break down to form reactive free radicals. So peroxynitrite is not a free radical, but it breaks down to form reactive free radicals. And these all then produce oxidative and nitrosative stress. And all of these things then come in to affect pathophysiology, to produce pathophysiological effects. Okay? But in addition to that, calcium signaling is extremely important in our cells. And so you have excessive calcium signaling working not through nitric oxide, but through other mechanisms. That also can produce pathophysiological effects. And so I'll argue that all these things, in fact, are involved in producing various kinds of responses that have been repeatedly reported to occur following exposure to these fields. Okay. So let's go. Okay. So this is Arthur Pilla's study where he talks about that, the therapeutic mechanism. And so I'm not going to have time to talk about it, but you can look at that paper. Okay, and these are several papers that I've published that I'm going to be referring to, and they include the first paper, which I mentioned before, the 2013 paper. This paper was included on the Global Medical Discovery site as one of the most important medical papers of 2013. And then I published a letter to the editor about therapeutic effects that was published in 2014. And then this one, this other study just came out just a few weeks ago. And this has quite a number of things in it. I talked about the voltage sensor properties to you before, but there are a lot of other things in there, including a critique of the Canadian report, which defends the claim that all you have to do is worry about thermal effects. And so we don't have time to talk about that, but I think there's a detailed critique in there which you may want to look at. By the way, this paper, this 2015 paper and the 2013 paper are both available on PubMed at no cost as full text articles, so you can download them at no cost. And so it costs me some money, but it doesn't cost you anything. Okay, so let's talk about health effects here. And so each of the things that I'm going to talk about, there are multiple studies that have reported these over and over and over again. The scientific evidence is very strong on each of them, but obviously there are people who are acting as spokesmen for corporations or for corporate organizations that will argue against them, and no doubt they will continue to argue against them. None of these can be explained by heating. All of them can be explained by VGCC activation, and all of that, of course, argues that the safety standards are nonsense. Okay, so let's go on and talk about some of these. One of them is oxidative stress. There's something close to 1,000 papers on oxidative stress following exposure to microwave fields, and that, of course, can be produced by excessive peroxynitrite and consequent free radical formation. And so there's that sort of thing. Some of my lines, by the way, got cut off of here. Single-strand breaks in cellular DNA and also double-strand breaks in cellular DNA occur and have been reported widely. They can both be produced by free radical attacks on the DNA, and there's an extensive literature on that. There's cancer, which of course is now becoming more and more clear that cancer is caused by these fields, and there are a number of mechanisms that I think are involved. And let me just say, when you have cancer produced by what's called inflammatory carcinogenesis, that when you have inflamed tissues, you get cancer occurring at higher rates in those tissues than in non-inflamed tissues. This has been known for decades. And the mechanism involved is this mechanism, which I describe here. And so I'm arguing cancer in response to EMFs occurs in very much the same way, okay? And so because all these things can occur as downstream effects of the VGCC activation, okay? So that's important. Breakdown of the blood-brain barrier has been reported, and peroxynitrite is known to be able to activate matrix metalloproteinases, MMPs. And so the blood-brain barrier is formed basically by tight junction proteins that tie the cells together in the vasculature in the brain. And so what happens here then is that when you've got a lot of peroxynitrite, you get much more activation of these matrix metalloproteinases. They then degrade these proteins that form the tight junctions fall apart and everything gets leaky, okay? So that's how you get the breakdown of the blood-brain barrier. There's similar things, by the way, that go on in the gut, and which I know some of you are very interested in. Male and female infertility, and I have a slightly longer thing on the next slide here. And these are, you get induction of there are a number of mechanisms that are implicated here that can have roles in male and female infertility. Male infertility, by the way, has been documented much more extensively than female infertility because it's much easier to study. Okay, but both of them occur based on various kinds of studies. And therapeutic effects we've already talked about. There are diverse neuropsychiatric effects of these fields. This is not widely known, but it is widely documented. And so there's a huge amount of documentation on this. And so you get diverse, and that includes depression, but many other things. And the VGCCs have very important roles in the nervous system. The nervous system seems to be the most sensitive organ in the entire body. And not surprisingly, because there are lots and lots of VGCCs throughout the nervous system, they have very important functions in the function of the nervous system. So you get melatonin depletion and sleep disruption, and that can occur through excessive calcium signaling, basically. Cataract formation has been widely argued by the defenders of the current safety standards to be caused by heating. It's not true. And you can actually see this in my 2015 paper where there's extensive documentation of it. But basically, cataract formation is known to involve excessive intracellular calcium under a wide variety of different circumstances. This has a key role in the process. And so you get excessive calcium signaling, and you also have a peroxynitrite oxidative stress role in formation of cataracts. So cataracts are clearly are not produced by heating. They're probably just another VGCC activation mechanism. And there are effects on the heart. And let me say, I think these effects are direct effects on the pacemaker cells in the sinoatrial node of the heart. Okay? It turns out the pacemaker cells in the sinoatrial node of the heart also have very high densities of these VGCCs. So they are very sensitive, and they can then produce tachycardia, but also bradycardia. So tachycardia is fast heartbeat, bradycardia is slow heartbeat. You get both. You get arrhythmia. Arrhythmia often leads to sudden cardiac death. We have an unexplained epidemic of sudden cardiac death in young, apparently healthy athletes dying in the middle of an athletic competition. I think the probable mechanism is EMFs. That has not been demonstrated. Okay, there has not really been much in the way of study on that, but it's an obvious mechanism acting through the process that I just talked about, where the pacemaker cells are highly sensitive. And let me say, there have been animal studies done on these effects, some of them going back to the 1960s, okay, on effects on the heart. So, okay, so basically all these things, as I've said, can be produced as a consequence of microwave action through the VGCCs, leading to both therapeutic effects and a wide variety of pathophysiological effects. And so as far as I can tell, the VGCCs are the only target that we're seeing of these fields at this point. Now that doesn't mean that we're not going to find one later, but we also know, as I've already said, why these VGCCs are so sensitive, because the voltage sensor is so sensitive. And so that's an important point. Okay, so Now, these are not the only pathophysiological effects that these EMFs can produce, but they're among the best understood, and they also give you some idea of the breadth of the effects that one is seeing here. And so, so simply based on the things that we listed in that table, we can say that fields are attacking the four things that we most value as individuals and as a species. And those include our health, our brain function, the integrity of our genomes, so when you attack the DNA, and the ability to produce healthy offspring. And that in and of itself, of course, is quite chilling. But believe it or not, when you start looking at some of the details, things get much worse. Believe it or not. I mean, okay, so let me talk about, so what I've been talking about, and let me just say none of these are proven in the sense that you can argue, but all of these are highly plausible in my judgment. And I think that at least four of them are quite probable. And each of them have horrendous implications. Okay, so I've put out 5 worst-case scenarios. And one of them is that the autism epidemic is probably caused by EMF exposures, but that chemicals also have a role. And I presented this as was already discussed in the introduction at the Autism One meeting in Chicago. And I discussed 32 types of evidence supporting a pathway of action for microwave EMF exposures, acting through disruption of the synapse development in the developing brain. Okay, and so this is a figure which I used in that talk, and I'm working on a paper now that hopefully will get all this published. And basically what you have here is microwave and lower frequency EMFs, of course, activating the VGCCs, giving you increases in intracellular calcium. And intracellular calcium then, and when it's excessive, disrupts each of these 4 mechanisms that are involved in the formation of the synapses in the developing brain. And so each of these are known to be calcium regulated, and so they are disrupted both by excessive calcium and also too little calcium, and also inappropriate levels of calcium. So the calcium levels have to be regulated appropriately in order to produce the proper formation of the synapses. And so it's not at all surprising then that we know that the connectivity in the brain of the connectivity of these neurons is severely disrupted in autism, and that's been argued based on various kinds of evidence to be caused by disruption of the synapse formation in the developing brain. And so we think that this is a key sequence, and as I said before, there are 32 distinct types of evidence. I'm not talking about 32 studies, I'm talking about types of evidence that document this sequence as not only occurring but having key roles in causing autism. Okay? Now, it's also true, however, that chemicals can act through excessive NMDA receptor activity, which also then produces excessive intracellular calcium. These two may well act synergistically together. And so I'm not saying it's all EMFs or all chemicals. I think there are roles in both. My judgment is that probably the EMFs are more important than the chemicals, but we clearly need to avoid, if we're gonna get anywhere in bringing this autism epidemic down, we have to greatly lower both of these. Okay. And now, of course, with EMFs, we're running as fast as we can in exactly the wrong direction with ever-increasing exposures. So, so that's, now, the most recent estimate of the autism incidence in the U.S. was 1 birth in 68, or about 1.5%. and the autism estimate worldwide is has been estimated at about 1%. The U.S. figures are based on births that occurred in 2002 and were assessed in 2010 and 2011. And so, first of all, this is old data in a sense, because we're talking about births that occurred 13 years ago now, right? And with the ever-increasing incidence of autism that we've seen over the last 35 years, about 150-fold over the last 35 years, over another 13-year period, you'd think you'd get much higher levels. So it's quite possible that the levels of people born in this year may be vastly higher. And based on the rate of increase that has been studied a lot in California, it could well be the case that births in 2015 we might be up to something like 1 in 4. Okay. I mean, this is speculation, but it's reasonable speculation. It tells you what an absolute horrendous situation we're in. Let me just say that autism, of course, is not the only thing that's going on here that's increasing dramatically. ADHD is also increasing dramatically, probably via a very similar mechanism. And it may well be the case that at this year, the kids that are being born this year, at least the males, and males are more sensitive, more susceptible than females for whatever reason. It may well be that the males born this year, all have neurological defects, and it's quite possible. Yeah.
Environmentally or in utero or both?
The conventional wisdom, which I think, okay, so the question is where are these exposures coming, where do they work in this process? And the conventional wisdom with autism is that the exposures that are important are perinatal, both late prenatal and early postnatal, okay? So they occur over that entire time period. And there are a variety of types of evidence that support that view, and so I think it's right. And so basically, we need to control we need to control the exposures during that entire time period. Let me just make a couple of comments here. So first of all, I think something that I think everybody knows. Each autism child places absolutely extraordinary stress on the entire family. I mean, the cost, not just economic cost, but the cost to people's lives, to what's really important, is just stunning. And and so an increase to something like 25% is just incredible, Now, as I say, that's speculation. We don't know what it is. We won't know what that figure is probably for another 10 years, okay? But what we do know is that this thing has been going up steadily, year after year after year, decade after decade, for at least 35 years now. And that greatly parallels the increase in microwave EMF exposures. So, I think that's, it's really stunning. But the other thing I wanted to mention, again, is that there are other neurological things that are happening and becoming more common, and ADHD being the most obvious of them. And so it may well be that, every male child, almost without exception, this year might have one or the other or both. And in females it'll be lower, but what's that going to do to our society? This is absolutely stunning, right? Now, again, it's speculation. This is not proof, but it is a reasonable presumption at this point. Okay, now, worst case scenario number 2, neuropsychiatric effects of microwave and EMF exposures. And so this is a list of neuropsychiatric effects that have been reported repeatedly in response to these field exposures. And okay, so microwave exposures, various types. You get sleep disturbance, insomnia, headache. Those are the two most common. But there are also many other things that are very common. Depression, depressive symptoms, fatigue, tiredness. That's not surprising. If you can't sleep, you're going to be fatigued. There are a series of dysesthesia. So here you have sensory dysfunction of several different types that occur. Concentration, attention, cognitive dysfunction. So many things that we think are important require effective concentration. And these people have huge problems with that. Memory dysfunction, dizziness, irritability, loss of appetite, restlessness, tension, anxiety, stress. All of these things, of course, are related to each other. So when you look at these things, and these are all, you talk to psychologists and psychiatrists, they'll tell you, oh, we see these, increasing tremendously. I don't know there's any good literature on this, but, you talk to people who are out in the field, and they tell you, yeah, these things are going up at huge rates. Now, if you have most of your population suffering from these things, and we may well be going in that direction with this ever-increasing exposure that we have, what the hell does that do to us? It's just almost incomprehensible. Okay, I don't know. And when you think about weird things that have been happening in recent years, almost all of them you can think of as being consequences of this. Let me just mention one other thing. One of the things I talked about just very briefly at the Commonwealth Club that two other people mentioned as well is that there is some evidence that these fields also cause loss of empathy. Now, loss of empathy is the classic symptom of sociopaths. Are we producing a society full of sociopaths? And if so, it's not a society at all. So these things are actually truly extraordinary. This is something that this cannot be ignored. Okay, so this is obviously a stunning and incredible thing, the neuropsychiatric effects. There's also issues of male and female infertility. I'm going to go through these very quickly because I've already run out of time here. The most extensive evidence is on male infertility being caused by microwave exposure. There is also very extensive evidence on lowering of sperm counts and sperm quality essentially worldwide. There's been a decline in sperm counts and sperm quality, unexplained. And so this may be the explanation, obviously. There are other effects that have also been documented on female infertility and also on increasing spontaneous abortions. And these then can be caused by these exposures. And there was an interesting study that was published something like 18 years ago by Magras and Xenos, which basically showed that if you took mice and you put them in two different areas in an antenna park. So these are all areas that are well within safety standards. So nothing should happen. And these mice, it doesn't take very long to go through gestation in a mouse. So you can go through a couple of pregnancies and get. But if you leave these mice in these fields for either about 2.5 months with a higher intensity, or about a little less than 5 months with a lower intensity, they become completely sterile. Now, you know, this is quite stunning. So you know, we know, as I said, we know we're having effects on sperm counts and sperm quality throughout much of the world. It's quite possible that we're going to become sterile, and you know, and reproduction will plummet, and what's that going to do? Okay, so let's.
It'll resolve the population problem.
Well, it'll do more than that. It's not going.
We're gonna change the batteries, so.
Oh, I
I'm
see.
so sorry.
Maybe you can you do that? Can you change the slide? Yeah.
Yeah, she can do it for you.
Yeah. Okay. So, so here's another one, and this has to do with genotoxic effects. We know that these fields are genotoxic. We know they damage the DNA to cells. We know from what I just discussed before that there's substantial impact on the germline cells. And so if you impact the germline cells, what's going to happen? Well, you're going to have lots of mutations passed on in the next generation, or not passed on because it kills them basically. And so there have been 3 studies done on germline mutations following microwave EMF exposure. They all show increases. It's been estimated that all we have to do is increase our mutation frequency by about 2.5 to 3 times what has been the normal level to make us go extinct. Okay. Now, so, okay. We're say scenario number 5. There's an epidemic of premature Alzheimer's which is unexplained. People coming down with Alzheimer's disease is age 30, 40, or even earlier than 30. You know, and this has been going up at a huge rate. I mean, this was basically unheard of a few years ago. And we know that epidemiological studies have shown that occupational exposures to extremely low frequency fields, which, as I said before, work very similarly in our body to the microwave fields, is associated with increased Alzheimer's. These are old studies, but they look quite solid. We have high levels of intracellular calcium, which has important roles in Alzheimer's. That's been extensively documented. And there's this fascinating study that was published on rats, where you could expose young rats to a series of short pulses of microwave EMFs. And by the time these were middle-aged rats, they had all the characteristic features of Alzheimer's disease, including high levels of the amyloid beta protein, cognitive memory impairment, oxidative stress in the brain. And so, you know, and this was published in this paper here, Archives of Medical Research. And Alzheimer's typically has a very long latency period, something like 25 years. It's possible that what we're seeing in terms of increased levels of these very early onset Alzheimer's might be due to the exposures that we had 25 years ago, which were vastly lower than they are now. Okay? Now, if that's true, and I'm not saying that it's necessarily true, if that's true, then we will certainly have a gigantic increase in early onset Alzheimer's occurring over the next 25 years, even if we stop exposures tomorrow. Okay? Now, again, I'm not saying this is true. What I am saying, all these things are plausible. Okay? And I think most of them are quite probable. So, anyway, so you know, that's okay. So that's the current epidemic. Yeah. So, you know, this is you know. So how do you look at this as a whole? Well, you know, based on the we may have high levels of infertility, birth rates may drop, and I think. I think what we have here is a number of types of evidence which argues that the effects of these fields increases with the length of time of exposure. okay? So you have a particular level of exposure, typical pattern of exposure, and when you are exposed or when an animal is exposed for longer time periods, things get more and more severe with time, okay? And there are several different types of studies which have been made which argue for that, okay? But what we're seeing now is not a constant level of exposure. We're seeing ever increasing levels of exposure. We don't have the foggiest idea what that will do to us. But we can be assured that it's going to be pretty bad and maybe extraordinarily bad. So let's see. We have a question?
What is the depth of microwave exposure into the body tissue? Like, is it something that only penetrates a little distance and gets spread through the blood system? Or
You know, okay, so the question is how far does this go in the body? And I think, you know, there are studies that have been done on cattle grazing near cell phone towers, all at levels well within safety standards. And the calves that are born to these cattle tend to have cataracts, okay? So it clearly can penetrate pretty far in to get to the developing eye in the fetus of these developing calves. So I'm just saying empirically, these things have got to penetrate pretty far. I don't know that, you know, other than that, I don't really have very much insight into this. So anyway, so all these things, you know, we have, we have evidence for various kinds of cumulative effects, and those are just where you have a particular level of exposure. With ever increasing exposure, we don't have the foggiest idea what we're doing. We simply don't have the foggiest idea what in the hell we're doing to ourselves. But we know it's going to be bad. So, what's this? Yeah, well, that's what I just said. Okay. Okay. So let's look at plausible consequences of these things, okay? We may see a catastrophic decrease in fertility, 60, 80, 90% infertility, crash in new births. If mice can become completely infertile, that's not unreasonable. Large fraction of the children may be born, may suffer from various deleterious mutations in the genome, those that are born are likely to suffer from autism, ADHD, and other types of neurological dysfunction. People will develop large numbers of neuropsychiatric effects from further exposure to these fields. And if anybody survives to age 40, they're at great risk for developing premature Alzheimer's disease. So, you know, yeah, well, anyway. So, you know, that's sounds very much like hell on earth. I don't know what, you know. These are all highly plausible, let me just say, and perhaps highly probable. No doubt people will argue about that. But the lack of absolute certainty on the effects on these effects cannot be a basis for the lack of vigorous action. It's putting us at great risk it would be completely unacceptable. It's just, so we are in an almost literally insane situation. And that's what the biology says, okay?
We have good cell service.
We got good cell servers. Yeah, right. Mm. Yeah. Okay. I think maybe I better quit on this. The cancer issue I've talked about at some length in this recent 2015 paper on how cancer can be produced, and I think what's interesting is that a lot of the cancer effects are produced through free radicals. And ionizing radiation also works through free radicals to cause cancer, okay? So the argument that ionizing radiation is very different in fact breaks down here, because both the ionizing radiation and these microwave and lower frequency fields work to a great extent by producing excessive free radicals in the cells of our bodies. So that's another thing where people have given assurances, oh, it can't possibly be true, and they're wrong. Okay, so okay, we've talked about these mechanisms.
You have 15 minutes.
I've got 15 minutes, but we got questions
No, well,
with the
four
question.
questions.
Okay. I was going to talk about EHS. I think that EHS is very similar to MCS in the sense that both of them involve excessive intracellular calcium. The chemicals act through the NMDA receptors. The EMFs, of course, are working through the VGCCs. They both produce excessive calcium in the cell, and I think the sensitivity responses are caused by that, by the excessive calcium through similar mechanisms, and I don't think I'm going to have time to talk about what I think those may be. So, but this is a series of similarities. So basically, okay. I think what I need to do is go to the end here and.
You can summarize, that's important to us, the EHS, if you can summarize that for us.
Well, I mean, what I think is that the microwave EMFs, microwave EMFs are quite active in activating VGCCs in the brain, but also in other parts of the body. And when you have this high level of activity and you raise intracellular calcium through exposures and produce nitric oxide and peroxynitrite, you can start what I call the no-no cycle. And this in turn will make those areas more sensitive, okay? And there are also effects on the neurological sensitivity, neurosensitivity, which is caused by what's called long-term potentiation, which is stimulated by both the VGCCs and the NMDA receptors, okay? So you get changes in the sensitivity of the synapses in the brain through these mechanisms. There are also regulatory effects, which I think are important at the cellular level. And some of those involve protein kinase C, some of them involve protein kinase A. Both of those increase the sensitivity of the VGCCs. And so this is a direct effect on the VGCCs, which makes them more sensitive. And so I think that's involved in the EHS as well. Okay.
Make up 70% of our clients.
70% of your clients are EHS. Okay. And so, okay. So, yeah. Okay.
Very important.
Right. Okay. Let me just say that what I proposed is a reasonable model. Okay, I'm not saying that this is proven, and I'm sure that it's actually more complicated than what I have in my slides. But it gives you some idea about how these things fit together, and it also gives you some idea that all these claims of there can't possibly be a physiological mechanism are nonsense. So in both of those, you also get with the EHS people, you get cardiac sensitivity. In many of these people, not in all of them, but in many of them, and that's probably produced through these the direct effects on the pacemaker cells in the heart. So, okay, I have a critique of the Canadian report in that paper. Look up the paper, and you'll see many different flaws in that report. I don't want to say anything more about that here. Overall conclusions. Okay, so this is the summary. Heating and thermal paradigm of microwave and lower frequency EMS should be replaced by VGCC activation. We have 7 types of evidence, each of which clearly shows that VGCC activation is the predominant mechanism for the action of EMS, possibly even the whole mechanism in mammals. Okay. Number 2, the voltage sensor of the VGCCs has physical properties that predict that it's exquisitely sensitive to these low-intensity EMFs. 3, Large numbers of repeatedly reported microwave health effects can be understood as being caused by downstream effects of VGCC activation, and you've already seen these, so I'm not going to repeat them. And 4, it can be seen from the above that low-level EMF exposures attack each of the 4 things we often most value as individuals and as a species: our health, our brain function, the integrity of our genomes, and our ability to produce healthy offspring. 5, with ever-increasing exposures to EMS, 5 worst-case scenarios each suggest that such exposures can rapidly lead to the crash of human civilization. And 6, the mechanisms of EHS may be similar to that of MCS. And 7, biologically based safety standards must be developed. The best way to do this is to look at the response of cells in culture to EMF, cells that have high densities of VGCCs. I mentioned that briefly earlier, and I think it's a key point in terms of what we need to do. Thank you.